right. Welcome back, everyone. Next, we have Bioxytran Inc. They are an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galactin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They're currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug being developed to treat stroke, which is supported by an FDA-approved device that detects tissue oxygenation. So due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. There is a third platform technology that is capable of treating cancer metastasis. So that is so much. I want to bring on Dr. David Platt. So happy to have you on our Emerging Growth Conference, Chairman and President and CEO. Happy to meet the man behind the galactin science. So welcome, Doctor. How are you today?
Thank you very much. I'm good. Thank you very much.
Okay, great. The floor is yours. And just call me back when you're ready for questions.
Very good. So I'm happy to present Bioxytran, which is a biological oxygen transfer. It's a complex carbohydrate chemistry company. And I would like to start with a good forward-looking statement. It's a public company. And we'll go into the presentation. In this case, we'll start talking about those 3 platform. So the first one is a viral infection platform. The second one is the cancer platform. And the third one is how we deliver oxygen with the carbohydrate. All of them is related to complex carbohydrate chemistry. And it's we're utilizing the mechanism of action is going by galactin, or those receptors that recognize predominantly sugar molecules. So to understand what we did in each platform, remember this is a platform, which each one we can make many drugs potentially. So the first one is the antiviral drug. Remember that this is made of complex carbohydrate chemistry, and at that platform we achieved remarkable data, preliminary data, because it's only finished phase 2, but we're talking about completely by PCR, by measuring the amount of virus in a person in the clinical trial, we achieve of 100% elimination of the virus by PCR, and the symptoms goes away. Basically, we cure all our patients in 2 sets of clinical trial. We publish the data, it's peer-reviewed, and it's there. The second platform, which is about cancer metastases, there is a drug. We have a couple of drugs. One of them is drug candidate that we can get into human trial when we have more fund, and that one to eliminate cancer metastasis. I'll go later into details about that. And the last platform, which is related to how to carrying oxygen by using polysaccharide or sugar chemistry to carry oxygen to tissue. And we have an FDA-approved 510K device and technology with extensive research behind it, and obviously approved technology, as I said, to detect every single oxygen molecule that mitochondria use in a cell. So we know exactly how many oxygen molecules are utilized by the brain, for example, so we can use it in tissue oxygenation and monitoring the oxygenation or delivering of oxygen. So we'll go to the first platform, and I'm emphasize the word platform because we can make many drugs, or we have one that we did. We're talking about galectin as a protein to recognize sugars. This is I would say, late science discoveries, only in the 80s, and other chemistries. And when we did that, I want to emphasize in this case what we did when we utilized that recognition to those receptors. We look on viruses, for example, a virus, and the virus, it's clearly, from our perspective, require the galectin or those protein receptor to recognize human cells. So that's number one. Number two, the galectin antagonists that we make, or those sugar structure that we're creating and design, supposed to block the receptor. And the third possibility, the third issue here, that we have an extensive IP protection here, because this is a unique chemistry, very complex chemistry, and it very precision here of the chemistry create that recognition. So the question is how we do that. For a non-scientist, and the excitement about what we have is that we take if you look on number C here on the slide, you can see the galectin, that's the receptor, that's a protein receptor, and you can see that piece that I show here, it's a sugar that it's recognized the galectin. So we take the galectin, and there is what you call galectin signature to each viruses. And you take that galectin signature, which is several galectin, and you, by NMR studies, nuclear magnetic resonance, we in vitro, and first of all, it basically on the computer, and later in the experiment, in the NMR, we can actually design the right structure to recognize those specific galectin, which I call galectin signature for a specific virus. And it's very similar to the early century when Paul Ehrlich was talking about, he discovered the drug for syphilis. And later with Fleming, with the antibiotics penicillin, there is a specific antibiotics for each bacteria, which Paul Ehrlich, by the way, call it the silver bullet. Here we have a situation that we can design a specific silver bullet for each virus. It's very clear that that specificity, we know in advance that we can eliminate, and then we do it in vitro, that we can eliminate the entry of the virus into the human cell. So after we did that, and we manufactured that, and we build a clinical trial, this is all under the FDA, you can see in phase 2, and we didn't compare it to a head-to-head in clinical trial, but if you look on the data, we're talking about comparing oral delivery of our drug, which is, we call it Prolactin M, to the known drug, oral drug of Paxlovid of Pfizer. So we are superior by definition, because in 7 days, that's what I'm talking in the early about what we have. We show a complete elimination by PCR compared to many other days by Pfizer's drug. And remember, this is not a head-to-head comparison, a different clinical trial, but the magnitude here that in a few days we eliminate the virus, and all that based on what you call classical drug design and synthesis prior to an optimization. The word here is to remember is optimization. We can optimize for every virus, we can optimize its own antiviral drug. So to summarize what we have here, prolactin M, which is our oral drug right now, it's in clinical trial. It doesn't affect by mutation, simply because the drug is what I call the galectin, are a conservative receptor, and they don't change in the virus. There's no the mutation in the virus for those receptors is very low. There's low limitation for any patient simply because carbohydrate chemistry are very safe for at least the drug that we do. There's no limitation of which patient, child, pregnant, or elderly. And we tested that. It obviously we showed there's efficacy, and we are now, if you look on the slide, we are actually in stage 2/3. We're doing the pharmacokinetic, or how much we need and how long. It's called what you call dose response. It's not just we did it in one drug and okay, with success, maybe it's not a coincidence. The same way I explained that we can design the chemistry, in this case, we did it again. We published that, and this is in shingles. And we're talking about taking the blisters. We did it in one patient because of a fund limitation, but we did it in one patient, and we described the results, which show that we can get rid of the blisters literally in hours, so we affected. So here is another situation how a precision in the chemistry toward as an antagonist to the galectin specific to the virus the shingle virus create a very strong clinical data outcome. So let's summarize what we have here. We have a platform for the first time, which we call it's anti-glycovirology. Remember, this is a new science. Anti-glycovirology banner. This is a new science in virology that you can design the same way you design in antibiotics in the last 100 years. And there are 2,600 viruses around. So we can go and for each virus, based on the galectin signature for each virus, we can design by in vitro and by the mathematics and the AI and all the tools that we are using, which is a convention tools in drug design, we can synthesize a specific drug for specific virus, and we can give it orally or IV, depending on the circumstances, and we can eliminate any virus. That's the potential. Okay, go to the next platform. Remember, this is about recognition of sugars. Sugars are known to get into the cell. That's how it to design. I don't want to get into the mechanics, but sugars are the basic. There are receptors to recognize them and all that. So the next platform is the hypoxia platform. What we did in the hypoxia platform, we designed a drug that can. It's small enough, 5,000 times small enough to penetrate, for example, in case of ischemia or a clot in the brain. So what we said, okay, if that's the case, we can make that drug, why not to use it as to carry oxygen to a tissue under duress, for example, ischemic tissue in the brain in case of stroke. So let's go and let's understand how we do that drug. So what we did here, we took an erythrocyte, red blood cells. From the red blood cells, we separate the hemoglobin, and out of that, we took the heme, the part that connected to oxygen. And then we put the heme and we connect it to a sugar molecule, a polysaccharide, a specific polysaccharide that we design to carry that heme. Now we have heme polysaccharide. And now that's the drug, and we can make a couple of drugs from that, but let's assume drug number one, call it drug number one. So we take the drug and we can utilize that in, let's say, a stroke situation. Why? Because in the early stage of stroke, if you look on on this slide, there is what you call golden hour. And the reason is people get hurt from stroke situation when the clot, if it's ischemic, the clot block the red blood cells and the oxygen does not get into the brain, the part of the brain it's blocked. So we have this drug, which is 5,000 times smaller than red blood cell, can penetrate the clot and can deliver oxygen to the part of the brain that it's blocked. The nice thing, it's IV drug, it's solution, you inject it, the drug goes to the lung, pick an oxygen, carry that as red blood cell to the brain, go through the clot because it's very small. The clot is like a net, it's like a fish net, and you go to the holes in the clot, and it get into the part of the brain, and we save the brain from damage till the patient get into the hospital. Now, what's the problem? You cannot treat the patient before you look for MRI, through the MRI, if it's hemorrhagic stroke or ischemic stroke. And if you give a patient RTPA or those other clot dissolved drug, you can kill the patient. So the patient has no treatment, and we have an advantage with this type of technology to save all these patients. We have results, and the results obviously preliminary, and we know that we can oxygenate that as I described. And the question is, what we're talking here? We're talking here epidemic in a disease condition that there's no unmet medical need in the early stage with a tremendous amount of expenses. We're talking about, I think I'm modest here, about half a billion. Maybe it's close to a trillion. In terms of expenses worldwide. So this is the table that gives you some ideas and what we're talking about, the enormity of this potential for this drug to go and deliver oxygen to the brain. We did more than that, by the way. We actually, if we developed a way to detect and that type of drug that we're creating. And we should remember that there is no drug available to deliver oxygen to the brain in early stage. None. Zero. Zippo. What happened today, patient go to the hospital, need to be distinguished between hemorrhagic, I'm repeating that, and ischemic, and only then some of them getting the RTPA, which is the dissolving the clot, and other technologies. But it's from the fact that damaging the brain is there for every single patient. What we did also to lower the cost for FDA approval for such a drug, which is a require major clinical trial and extensive clinical trial with many patients, we also developed an FDA-approved device. We call it the MDX viewer. And that device will be able to detect the rate that we oxygenate the brain during the treatment early. So we can calculate how many oxygen molecules we deliver to the brain in any given time in real time. This is part of our unique capability in this area of research. The next platform, remember, we're still in the describing what we do in each platform. Then the next platform, it's about the cancer metastasis. And in the cancer metastasis, what there for many years, I'm doing the research in that area, and part of that in making all those antagonists related to cancer metastasis. And right now, over the years, there are many publications about that. I will just remind you where I'm coming from. I will go to the first, to one of the first slides that I have here. Okay. To understand, I missed that slides because I realized that as I'm talking about the presentation that it's very important for you to understand who am I? I mean, what I'm doing in this field. Many people don't know me, and first of all, I'm the one who first time expressed the galectin receptor. At that time, in the late 80s, I mean, there was no one in science knew what I'm talking about. I realized that early in my career, how important is this receptor? And to date, by the way, I call it galectin, and there are two textbooks that I created, an article. Those are the articles that I wrote first time, and I described that receptor. Since then, there are more than 4,000 publications about galectin. For my first time that I published that in 1992, and I call it galectin. And I'm very proud of that because it shows the understanding that it actually reinforced the understanding about galectin in this field. And my contribution to that field of utilizing the galectin as a way to creating drug, because it's in the end of the day we translate science to drugs. So to summarize what we have here, we have basically two platforms that we're concentrating right now, simply because fund restriction and the way what you call low-hanging fruits. So the first one is the prolactin with the tremendous data in the clinic, which will continue. And the second one is the BIXT25, which is the carrier, we call it the universal oxygen carrier, which we can utilize in stroke and Alzheimer patients, for example, where for toxicity purposes, we may use Alzheimer patients, and when we'll get into clinical trial. We're very close to do clinical trial on the BIXT25. What I want to describe in the end is something very simple, and what is the value of this company for new investor and old investors or existing investors. We have a very experienced team management in the company. The cost efficient activity is right there. We are doing it based on the experience And we're not talking about spending $200 million to make a drug. We're talking about a fraction of that. With many years of experience in manufacturing and clinical trial, we are very concentrated and we know exactly what we need to do to get from point A to point B. The science is obviously contain the risk situation and for investors. Also, there is many inflection point or value inflection point for investors in those three platforms. And right now we have several institutional investors potential investors who will take the company to the next level. The exit for this company is obviously a license. We are, once we get to the point that the proof of concept, I call it, the proof of concept, POC, the proof of concept is mature enough, then we will do a license for each technology. So describe the company in itself, we're talking about an OTC company, QB, fully reported, with 2,000 shareholders, with 175 million shares outstanding, and the management own and the people hold close to the company, insiders, holding 53%. So we'll go to the Q&A. Please, Anna, if you look on the chat, let me know what people.
Yes. Wonderful. Thank you so much. It seems like you have dozens of viruses that you could potentially target. So, Doctor, the question is, what do you believe is the most important virus to focus on, taking into consideration the clinical trial costs, efficacy, and the patient availability?
This is a very important question. Who asked that? It's a very important question. I think that the data we have now, the preliminary data in phase 2, speak for themselves. We're talking about a complete elimination of the virus in proof of concept clinical trial. We can take any virus. It will be, but we have the data. We have the data on one virus, so we need to concentrate on what we have. It's a winning horse. We're talking about curing all the patients, phase 2, from the symptoms, from the virus, eliminating the virus. And after 14 days, they all had antibodies, so they were protected naturally. That's a tremendous data. We should concentrate on what we have. Obviously, we can do other viruses, but that's a question of funding, and we will concentrate on this specific virus, and I'm super excited about it. We're talking about long COVID. It cannot get better than 100%, and
Right.
And this is something that I'm extremely excited about it. When you have a science, theoretical science, that it's translate in the clinic to 100%, we should keep doing that. And it's just a question of time to get it approved or to get a license for major pharma or collaborators. It's just simple as that.
Well, Doctor, lastly, we just have a minute or so. Quick question. When did you first find out that the galectin inhibitors were potentially useful in treating metastatic cancer?
My PhD, let's talk about two forms. I want to answer this question in two parts. First of all, personally, my mother died from breast cancer, metastatic breast cancer at age 43. So that's a personal loss. So I got attention to the cancer naturally. But my PhD thesis was about pectin. my PhD, it's in the 80s, was about pectin. So pectin is a polysaccharide from a cell wall. So I connect the knowledge about galectin, who recognize sugar. Those are lectins receptor that recognize sugar. So it can be in antiviral, it can be in cancer. My early interest was in cancer. So I was interested in that, and I find out that I can use my knowledge in polysaccharide to block cancer. So the result that I published early showed that I can inhibit cancer. So it's not a new thing. But the thing, it takes a long time, because to date, there is no, at that time, there was no liquid biopsy. So today I have the ability to continue that research and do a clinical trial on the molecules and get it approved. By inhibiting cancer metastasis, continue the work that I do all the years,
Well, this is a fascinating story, and it seems like a very exciting and life-changing journey you are on with Bioxytran. We're so happy that you came on to the conference and explained such important work that you're doing. So we would love to follow you along in the future with some updates, Doctor. Thank you so much for joining us today.
Thank you.
All right, we'll be right back with our next presenter.